Hey guys, what's up? It's Mr. Brotherton here. I'm gonna kind of do a really fast recap of protein synthesis. This is a very complicated process in biology where there's a lot of stuff happening. I'm gonna try and do it in 15 minutes because that's all I have. So let's get to it. All right, protein synthesis. This is all about how we make proteins. Proteins do everything in your body. All right, and how do we get there? How do we get to the point where we have a body made of all these different proteins, and how do we kind of pass that information along down the line, individual to individual? Well, it's because of DNA. So DNA is basically just a big recipe. All right, it's a big tangled mess of recipes for making proteins. Human beings have about 19,000 different protein making genes. We maybe have 23,000 genes or so. Some of those do other things besides specifically make proteins, actually make RNA and stuff, but that doesn't matter. And that has nothing to do with how complicated an animal is either, by the way, because grapes have about 30,000 genes, okay? So how does all of this work? DNA is basically just a long cookbook. Each gene is essentially just a recipe, and those recipes get handed down generation to generation to generation. So how do we get traits? Well, traits are just a bunch of proteins stuck together that make us look a certain way. All right, so proteins, once again, these are incredibly important because they do everything in your body. They are what puts stuff together. They're what transports stuff around your body. They're your hormones. They help you defend against viruses. They connect your cells together. Really important one that we're talking about in my anatomy and physiology class right now is myosin and actin. One that you guys have actually been exposed to, a protein that you're not aware of, is dystrophin. I had you watch a video of how those kids were living and the way their lives were similar from each other or different from your life. And actually that all comes down to one single mutation that changes dystrophin. And dystrophin is a protein that allows your muscles to stick to one another. And if you're missing that protein when you exercise, your muscles kind of break apart. So that's another one involved in movement. All right. All right, so let's get into the central dogma. How do we go from DNA to proteins? It's all about this process right here. DNA makes RNA, which makes protein, and that's two different steps. So DNA to RNA, that's known as transcription. RNA to protein, that is known as translation. So let's take a look at this. So transcription happens in the nucleus, translation happens in the cytoplasm. Now, why is that? The thing is, DNA is very, very, very precious. It gets passed down generation to generation to generation. Inside of this nucleus here, it's nice and protected. Okay, it has this cell membrane to protect it, then it has the inner membrane of the nucleus to protect it. All right, so we need to be able to read this recipe, but we don't want this recipe to get messed up or destroyed out here in the dangerous cytoplasm. So we make a copy of it, and that copy is known as messenger RNA. So the way I'm going to teach this to you is through a cooking analogy. This is my favorite way of understanding this process. So all an analogy is a way of describing something in terms we are more familiar with. Okay, so DNA is basically a whole bunch of cookbooks. That's how I think about it. A gene is just a recipe in that cookbook. And now imagine if you had some amazing cake recipe handed down to you from your great-great-great-grandmother. Every time you wanted to make that cake, you probably wouldn't want to get that cookbook out because you might get it dirty, you might get it messed up. So you wrote it down on a note card and you made a little copy of it that it doesn't really matter if it gets destroyed. So that's what messenger RNA is. It's just a copy of that super precious DNA, so we can take it outside of the nucleus and do something with it, okay? The next thing is that mRNA goes and it is translated into protein. So Pay attention to the words here. I know you guys don't think about words as much as we did. You should really take Latin. And I know that they took the analogies off the SATs, but maybe they shouldn't have. That's just my little soapbox, but here we go. Let's think about what this word means. Transcribe. So a scribe is someone who rewrites something. Okay, so we call this process transcription because trans means to basically move. If you think about the word transport, and a scribe is someone who writes. So we call this process transcription because all we're doing is we're rewriting DNA as RNA. They're basically the same language, okay, but we're just rewriting this one as this one. Think about American English and British English are the same thing, right? But we say things slightly differently. So in DNA, there's a T, so it's a T, G, and a C. In RNA, there's a U, a G, and a C. Well, the British spell color with a U, we don't. And maybe RNA is like the British, so it spells things with a U. So transcription, it's just rewritten. They're essentially the same language, but they just are slightly different variations because they're kind of in different handwriting. All right. This word translation, all right, just like you're translating something into a different language, so English to Spanish is a lot different than American English to British English. Now you're doing something entirely different. You're taking the language of nucleic acids and you're translating it into the language of proteins. All right. So there's a couple different types of RNA. RNA is a very cool molecule. Like I've said a million times, it's been around forever. So the first kind that you saw on that last slide is messenger RNA. Messenger, why? Because it's just a copy. Okay, its job is just to say, hey, this is what the DNA said. All right. Now these two types of RNA are really, really cool because they are made of RNA. They are made of nucleic acids, but they can kind of act like proteins. Okay, they actually carry out a job very similar to what a protein would do. But once again, RNA is much older than even proteins or DNA. So RNA is really cool because it can pretty much carry out all the tasks of life by itself. Kind of awesome. So messenger RNA comes out of the nucleus and it meets up with these guys, transfer RNA, which basically it's in the name. Okay, this thing just carries an amino acid. And a ribosome, this guy is the chef. This is who really cooks the meal. This is who really puts together our protein. All right. So messenger RNA, this is basically a copy of our recipe. Our transfer RNA, this is kind of just the sous chef or the prep cook or a line cook who their job is to just bring this thing right here, this amino acid. These are essentially our ingredients. So all this guy does is bring stuff to our head chef, and that is our ribosome. This is the one who really reads the recipe and puts everything together. All right, so transcription, what's our purpose? Basically carry the code that's on our DNA, turn it into RNA, which can be destroyed. It doesn't really matter that much, and bring it to our chef, who is the ribosome. All right, so what does that look like? We need to unzip our gene. We need to get in there and read our gene, and we need to make sure that all of our stuff matches up. So our RNA complement here is just matching up to our DNA, okay? So that way we get an exact copy of what's going on. So we're just using the same base pairing rules that we use for DNA, except now we're using them with RNA. So if you look here, like the C matches up with the G, this A matches up with a U instead of a T, this T matches up with an A, et cetera, and then this thing can go out into the cytoplasm. And then once this thing's done and it leaves, it can zip right back up and go on its way. All right, so here's a picture of what this looks like. So we have DNA all nice and happy, zipped up, safe, and protected. Okay, so now we have this DNA basically opens up. Okay, so this is this DNA, this is this side of the DNA, and now we have RNA coming in and making a copy of this. All right, and then eventually the DNA zips back up and the RNA is able to go out into the cytoplasm. So here's an example for you guys to try. So far, sequence is TACGCTA, GTA, CGATT. What would our mRNA be? Well, it's going to be the opposite. So it's going to be AUGC, etc. All right. So now let's go to translation. This one is a bit more complicated. I'm going to try and go through it as quickly as I possibly can. So translation, this is what's happening in the cytoplasm. So we still have our messenger RNA. All right, this thing is basically taking our recipe, and it is going to meet up with the ribosome and our transfer RNA. So what's this thing, a codon? Well, a codon is basically how we actually take that DNA sequence, read it, make sense of it, and turn it into protein. So DNA is just a bunch of ATs, GCs, whatever, strung out. That doesn't really help us get to protein. What helps us get to protein, though, is if we take a combination of 3 of those bases and put them together, now we have something that makes sense, because each one of these codons, you'll see here in a second, actually matches up with an ingredient in our recipe. So each one of these is like a word in our recipe. All right, so this has a meaning. UCA means something. It means, hey, there's an amino acid that matches up with me. All right, and this thing's job, our sous chef or our prep cook, its job is to go match or go grab that ingredient and bring it to the messenger RNA and to the ribosome that is our chef. All right, so a codon, just think of it as a word. It's basically a word. What does the word mean? It means, hey, I'm a type of amino acid. An amino acid is just the monomer. Remember, mono means one, mer means number. So it's a building block that makes proteins. You guys have probably heard of amino acids before. There's 20 of them, and you string a whole bunch of them together. It makes a big, long thing, kind of like DNA, except it folds all over on top of itself. Its complicated shape allows it to do its job. So this little black thing here, that's an amino acid. Anticodon sounds a lot scarier than it actually is. All it is it's the part of our transfer RNA that is able to read a codon. So a codon's the word, the anticodon is the opposite word, and it just has to match up to bring our amino acid. All right, so translation. All we have to do, read the instructions and make our protein. Okay, so this is where we actually cook our recipe. All right. Here's kind of the whole process laid out for you, but I'm going to show it to you on this slide right here. So if you imagine right now, this whole thing you're looking at would be inside of a ribosome, this big circular thing, okay? And what the ribosome does is it basically allows the RNA to come inside of it, the messenger RNA to come inside of it. Then a transfer RNA comes inside of it, and it matches up the mRNA codon here to our anticodon, and then it drops off an amino acid. I really suggest you watch the animations that I've posted because they are much, much more helpful to actually see what's happening. But each one of these comes and drops off an amino acid inside the ribosome. Then they all get stuck together with a bond, and that bond is what makes a protein. So here we go. This is how you actually read our genetic code. Okay, I always joke that GUAC is the key to life, but here is what's known as a codon chart or an amino acid chart, and this is how you actually read mRNA to make a protein. So every single gene starts with AUG. So if you look here, see how this says AUG? That's how the ribosome and the transfer RNA knows that there's even a gene there. Every single gene starts with AUG. That basically says, hey, start here, okay? And so what happens is it reads the A, so I'm reading the A first, then the U, then the G, and that brings an amino acid known as MET every single time. So then if we go back here, our next codon says UCA. So we go UCA. So then that next amino acid is known as SIR. Okay, what's really great about this codon chart right here is it's actually showing you that each one of these amino acids has a different shape. These are their actual shapes. So you can imagine you string hundreds and hundreds of these together, you're going to get a complicated protein with an entirely different shape. All right, so real quick, that was transcription and translation as fast as I could possibly make it. Please look at this. Please watch the videos because I know this isn't good enough.